And so that's what we're going to look at today is microbial relationships to systems management choices and production practices across a couple of different systems. There we go. So just to get started, so we're all on kind of the same playing field. We're all pretty familiar with soil physical and chemical characteristics. Things like soil texture, bulk density, aggregate stability, pH, nutrient content. Biological components are a little more nebulous. We know that they're important, but we don't necessarily have standards for what they look like in a healthy system or anything like that. What we're generally talking about for that are things like microbial biomass, enzyme activity, which helps with nutrient cycling, disease and pest resistance, and that kind of thing. The reason that I spend a lot of my time talking about soil biology is this right here. And that is that the soil biota, whether we're talking about bacteria, fungi, earthworms, beetles, all have huge impacts on a lot of soil functions. They influence things like aggregate formation and aggregate stability through the formation of things like glomalin and organic exudates, and the mycelia that actually physically hold soil aggregates together in the soil. They help with water infiltration and water storage. Helping to decompose residue actually contributes indirectly to nutrient cycling because they're taking all of that nutrients that are tied up in roots and plant material and releasing it back into the soil. We also have things like rhizobia and bradyrhizobia, which form symbiotic relationships with plants to bring nitrogen into the system where it's not readily available. And then there are groups of organisms that do this in non-symbiotic relationships that just help bring nitrogen into the system on its own. So there's a lot of reasons, whether it's an indirect or a direct relationship, that the soil biology is very important, whether we have the best assessments for it or not. This is what we look at when we look at the soil. And this is sort of where I came into it. You have your plant root growing into the soil, into the sand, silt, and clay particles here in these aggregates. You have a mycelia, fungal mycelia, making that symbiotic relation with the plant root, growing out in the soil, accessing nutrients and water that the plant might not have access to. You have things like mites, nematodes, and protozoa all living in those pore spaces that are helping to break down organic matter and cycle those nutrients through the system. Inside these smaller spaces where we find our silts and clays, we have our organic matter buildup, bacteria, and other organisms that are forming there as well. And so what we're trying to do from a conservation perspective is design systems that promote this kind of interactivity in the soil profile. And this next slide is one that actually came out of my PhD research working with my major advisor of sort of how we go about designing and assessing these systems. So the first thing is we have a goal. And our goal from a conservation perspective, we want more material going into the system, we want more variety of that material, and we want it to be biologically available, okay? So we're going to implement practices that bring in that, whether that's no-till production or reduced till, cover crops, mulching, leaving that residue on the soil surface if we can, because when we do that, we typically see increased number and types of organisms, so increased microbial diversity, and we also see turnover of that organic matter, which influences things like carbon sequestration and where did that carbon end up. We also see improved aggregate stability, which helps our soils resist erosion, increasing water infiltration, and that biochemical cycling where we see the release of those organic nutrients into inorganic plant available forms. Because the goal of all of this is that we want improved growth and productivity regardless of the system we're working in, whether we're looking at pasture land systems or we're in row crop production type systems. Now the thing is, and you've seen that this morning as well, all of this is site specific. What works for one person may not work for the other, and I'm going to show you some of that today. So one of the first projects I want to show you is actually work I did when I was going to school at Texas Tech in Lubbock here. So about 18 inches of rainfall on average per year, pretty low. And we were looking at comparing cotton monoculture systems, because as they say in West Texas, cotton is king, and that's what most people were producing in that area, to some long-term systems that had been implemented where they had producers that had grass-fed cattle, and then they also had part of their system in a row crop system. Now, sometimes they were actually grazing these, and then part of their system would usually be in cotton production as well. So the first one that I'm showing you, this had actually been in place in 13 years by the time that I got to West Texas. And what we saw is the organic matter and organic carbon in 1997 compared to what it was 13 years later when we collected samples, and you can see where we had that integrated crop livestock system, a 31% increase in soil organic carbon. Now for some people, 31% increase, that doesn't sound like a whole lot. It doesn't sound super impressive, but when we're looking at a system that, again, has very low rainfall input, they have part of it is in rotation for grazing as well as row crop and cotton production, it was pretty impressive for those soils. The other part of this is they found that they were using less irrigation water for these systems. They were putting out less applications of fertilizer and other resources as well to it, okay? So we wanted to know, so what this impact is having on the soil biological and the chemical components as well. Now we didn't have 1997 samples to be able to do that, but what we did have was a new project that had been implemented most recently, working with the Texas Alliance for Water Conservation and working with producers who were either looking at ways to reduce irrigation in continuous cotton or in these integrated crop livestock systems. And what we found is even in these systems that had been in place for, in some cases, maybe 5 to 10 years, we were still seeing that increase in soil organic carbon, depending on what system we were looking at, anywhere from 15 to 50% increase. And this last system, so these two were actually on research stations. This one was a producer operated system where he was grazing a quarter of a circle, and then the other quarter he had in row crop production that he would graze occasionally, and he was growing cotton, wheat, and corn throughout the year. And so he had some of the nicest soils that I had worked with in an area where moisture was particularly limiting for them. But what did that mean from a soil health and a soil conservation perspective? So one of the things we wanted to look at is aggregate stability. We measured it as a function of mean weight diameter, and I just put this up there to kind of show you what we're looking at is these aggregates here. What happens to the soil when it's disturbed, usually by a rainfall or some kind of physical event? How well or how cohesive do those aggregates stay stuck together over time? And for this method, what we do is we have different sizes of sieves, and we pass the soil through that sieve, and we measure how much soil stays on each size, and that gives us an average aggregate size. We can then average the amount of those aggregates, and it gives us what's known as mean weight diameter. So a bigger diameter means a higher aggregate stability. And what we found is all of our fields that were in annual crops clustered right here. And you can see that though we do have a range in organic matter, we're not really seeing an improvement in aggregate stability in those fields. They're not really seeing that cohesiveness of the soil particles. But when we move into our perennial systems, and those part of it, we do see not only increasing in organic matter, but increasing in aggregate stability. And in fact, when we look across the two types of management, we had almost a 3 times increase in aggregate stability in the perennial systems compared to the row crops. So that stands to reason we've got less disturbance, more organic matter going into those systems. It's helping to keep those soils more cohesive and held together. We also wanted to look at this from a biological component, which is why I'm here today. And what we're looking at here is we have, again, those monoculture cotton systems and those rotation perennial systems. And we're looking at the number of different species of fungi in this case. And you can see there's a big difference between our monoculture cotton system and those diverse rotational and perennial systems. So we're seeing a significant increase in just overall fungal diversity across those systems. When we then look at how that relates to something like soil organic matter, again, we see our monoculture cotton and then our rotation and perennial systems here as those numbers start to increase. So we're tying this again back to that increase in organic matter, an increase in fungal diversity in fungal communities, an increase in aggregate stability. When we look at a specific group of soil fungi, here we're looking at arbuscular mycorrhizal fungi. What we found is that in the surface, which is this line with the red dots, and even in this, what we're calling subsurface in this case, or about 2 to 10 centimeters into the soil, we do see that as we increase populations of our vascular mycorrhizal fungi, we also increase mean weight diameter or aggregate stability. So again, we're seeing all of those interconnectedness and the linkage between all of these different communities and organisms. The reason we focused on our vascular mycorrhizal fungi is not only do they contribute to this, but they also bring in these other relationships where those fungi make symbiotic relationships with the plants, and they bring in things like resistance to plant and root pathogens. They increase, their increased tolerance to things like salt and drought stresses, and increase access to soil plant nutrients. And our vascular mycorrhizal fungi are pretty cool because they have the ability to make that symbiotic relationship with a wide variety of plants, okay? As opposed to something like rhizobia, which are working specifically with legumes. So now I want to change gears a little bit. We're still looking at grazing land systems, but now we've moved to Louisiana and Mississippi. So I went from 18 inches of rainfall to 68 inches of rainfall in a year. It was a little bit of a change for me. This one actually came about because we were working with a producer named Cooper Hurst. He had been overseeding his pastures with winter annuals for about 9 years at that point, with a diverse mix of these. And this is just a sampling of what he was using at that time. Cooper would basically go in, see what he could get available, and that's what he made his mixes out of each year. His goals were to improve organic matter and soil health, and also, because we're in southern Louisiana, provide a forage that his cattle could graze pretty much all year round. And he was getting that from the system. Our goal initially was to try and relate what was going on below ground to what was going on above ground. Some miscommunications in the research world, which happened, means we couldn't actually do that, but we could still go in and see what was going on in his systems over time. And one of the things that really stood out to me was that we weren't seeing any changes in things like soil organic matter, total carbon, and total nitrogen. Now, in perennial systems, we thought we should have expected to see that, but what we hypothesized is these had been in place for about 9 years. Cooper had been managing it the same way year after year after year. So it's very possible we had basically reached a level of equilibrium in these systems as far as inputs of nitrogen and carbon. But what was really interesting is this dashed sort of purple line here, right here. That is nitrate nitrogen, okay? He was not adding any nitrogen to his system, okay? And in fact, when we asked him, because we saw that nitrate nitrogen was decreasing, what was his fertilization plan? He didn't have one. He had not seen any decrease in yield from his forages. He hadn't seen any nutrient deficiencies. So he saw no need to be adding nitrogen to his system when he was grazing, which I left that part out. He was rotationally grazing. He was in there any 12 to 24 hours moving cattle through his fields. So we were a little confused. He's not adding nitrogen. He's not seeing the need for nitrogen. And we're not seeing any changes here, but we're definitely seeing losses of that plant available nitrogen. So it's happening. So we started looking at the data, and we started looking at organic matter and microbial biomass, and we saw that even though organic matter wasn't necessarily changing over time, he did have variability across his fields. And you can see anywhere from about 1% up to almost a little over 8%, depending on where that sample was collected. And as we look at the microbial community, we see that total microbial biomass is actually increasing as we see increasing organic matter, which again makes sense. We have that organic matter acting as a food source for the microbial community, and it's feeding into that and helping that microbial community grow. And then we started looking at how that related to nutrients, and in particular, inorganic nitrogen and nutrients like soil test phosphorus. And it's not as clean of a relationship, but we do still see that as total microbial biomass increases, things like inorganic nitrogen and soil phosphorus availability are also increasing. So our hypothesis was, okay, we have this source of inorganic or total nitrogen. The microbial community is actually helping to break down that organic nitrogen and release it back into the soil as inorganic nitrogen. We're just not capturing that because we're sampling once a year. Unfortunate part of research, I am a little bit limited by time and money. I can't sample every time I want to. So we think that's what's happening here. And one thing to back that up is looking at this. Now it says nagase. This is an enzyme known as N-acetyl beta-D-glucosaminidase, which trips up every single grad student I've ever had. But basically what it is, it's an enzyme that helps with carbon and nitrogen cycling. So it's one of those enzymes that helps break down organic nitrogen and release it back into the soil in an inorganic form that is plant available. And we can see as our microbial biomass increases, the activity of that particular enzyme also increases. So as we see organic matter increase, we grow our microbial community. That microbial community is releasing the enzymes that we need to break down that organic matter and helping us to cycle those nutrients. And just to show what we're talking about for this is actually glucosamine, which is the basic chemical makeup for what it's breaking down. And you can see there's that nitrogen group that's being released. It's also very interesting because this glucosamine is a main component in chitin, which is related to things like the exoskeletons of insects and spiders, as well as fungi that we find in the soil community. Now, like I said, we weren't able to answer all the questions we wanted to with this research. Oops, sorry, I got a little happy with the button. So we went back to the drawing board and we wanted to design an experiment where maybe we could actually relate what's going on below ground with what's going on above ground. So we looked at a variety of treatments for cool season annuals. We had legumes, grasses, brassicas, mixes of all of those. We threw everything we had planted into one bag and just planted that to see what would happen, as well as a ryegrass and a check plot. We also wanted to look at harvest frequency. Now, I want to add though, unfortunately we were not able to actually graze this, so we're missing that component of the animal waste going back into the system. But what we are replicating is the removal of that biomass, either at 4 or 8 weeks, and then at the end of the season, just mulching everything and letting it grow back into it. And then to add another level of complexity to it, we did it at 3 locations. We have one here in Southwest Mississippi, one in Northwest Louisiana, and one down here in Baton Rouge. I was incredibly excited for this. It's the closest research trial I've ever had since I moved to Louisiana. Everything else I have is a minimum of an hour-and-a-half drive. So I was also a little brokenhearted. We had the best stand of white clover I've ever seen, even though we never planted white clover once in that field. So that kind of goes back to that whole thing of things are site specific. Things are going to respond differently in different locations, okay? So most of what we're gonna see today are for these northern locations. But just to take a first look, how are we affecting biomass? So here we had our grass, our legumes, our grass and legumes, annual ryegrass, and our control. And you can see that in both locations, we are increasing overall total biomass or total forages from that location. Now, we're also analyzing this for forage quality. I'm not a forage specialist, so I don't have that data with me today, but we are working on getting that information. But we are seeing that having those cover crops are benefiting the overall forage production from those systems. When we start looking at how is that influencing things like soil properties, again, here, because these are newer systems, we actually started these in 2017 and we're going through, we are seeing that expected increase in total carbon and total nitrogen. But interestingly, again, we're seeing that stark loss in nitrate nitrogen in the system as well. When we look at how that was reacting to things like that glucosaminidase enzyme in the breakdown, we see that regardless of how it's being treated, we are seeing increases in the production of that enzyme, with the most increase seen where we're mulching it. And essentially mulching, we were going in, they were mowing it and leaving all the biomass on the surface. So we're not taking anything out of the system. So it makes sense that where we're having the most inputs, we're seeing the most response from the microbial community to put those resources back into the system. And at Hill Farm, regardless of what we put in, we were seeing increases in that nitrogen cycling enzyme as well. Ben-Hur, a little bit of a difference. We were seeing, again, mulching in general gave us higher glucosaminidase activity. But what was really weird here is at 8 weeks harvest, we saw increases in grass, legumes, and grasses, brassicas, and ryegrass. Legumes, not so much, which was completely opposite of where we were mulching and we were doing 4-week. And I'm still not real sure what exactly is happening there, particularly at this 4 weeks with the legumes. I'm hoping that maybe something we see in the forage data will help support what we were seeing. And then no microbiology talk is complete without at least one diagram like this. And basically what we're looking at is the fungal or the biological community in these systems at the hill farm. And so each one of these points represents all of the fatty acids extracted from one single soil sample. And what we wanted to know is how does this sample compare to this sample and every other sample here? When two samples are really close together, they're pretty similar. When you have this sample compared to this sample, they're very, very different. And I know you guys over here can't see what I'm circling, so what I'm talking about is like this sample up here compared to this one here is going to be very different than these two samples are. And what we found is that when we were harvesting every 4 weeks, we typically saw higher populations of things like AMF and protozoa. When we were harvesting at 8 weeks, which is the yellow one, we were seeing higher populations of fungi, saprophytic fungi, which are fungi that feed on dead material, and gram-negative bacteria. And then when we were mulching and leaving that material behind, we saw higher populations of bacteria, specifically gram-positive, and actinomycetes, and eukaryotes, which usually correspond to things like algae and maybe some plant root exudates in that case. But so we weren't seeing big changes because of the above ground biomass, but how we treated the biomass, how frequently we removed it from the system was having a very large impact on the microbial community in that area. To change gears just a little bit, I also want to talk about row crop production. This is a trial that actually was started by Dr. Josh Lofton and Beatrix Haggard in 2013 or so, and that I had the opportunity to take over upon being there. And this is in 5 years into the project. I want to show some of the data from that. But these systems have been in no-till since 2013. I keep getting people from the farm station trying to get me to turn them over, and I won't do it. But I have to keep fighting for it sometimes. And then we've been planting cover crops, and we've also been overlaying this with nitrogen application rates for different production practices. So just to start out to show, we are seeing nitrogen credits from legumes. You can see here, these are fields that had not had any nitrogen applied, and we were seeing almost a 50% increase in yield. So we're seeing a nitrogen credit from the legume cover crops. Obviously, not enough to get up to where we want to be, but that's pretty impressive there. At 80 pounds per nitrogen for this corn field, we are seeing our highest yield, and we're maximizing our yield at 80 pounds under legumes, and almost 20% higher than we were seeing in our grasses and our fallow treatment. And when we looked at this for economic optimum nitrogen rates, we actually were getting a 40 pound per acre nitrogen credit from those cover crops. And that for us was pretty impressive because we're terminating our cover crops usually February 1 to try and start planting as early as February 15th into the 1st of March. So our cover crops aren't getting a lot of time in the ground going into corn production. From a soil perspective, we were looking at nitrate nitrogen levels, and what we saw was really interesting. In the fall, after harvest, we're seeing sort of that residual nitrogen left over from the corn production. In the spring, after the cover crops have been growing all winter, we don't see that anymore because those cover crops are scavenging that nitrogen and tying it up in their biomass. When we look at how this is influencing things like enzyme activity, I don't know how well it shows up. There's the light blue bar. That is a carbon cycling enzyme, beta-glucosidase. It's breaking down things like cellulose and hemicellulose in the soil. And then the dark one is that glucosaminidase enzyme. And you can see over time, we're seeing increases in that production of that enzyme. What's interesting is things like this glucosidase enzyme, the lighter blue bar, it's actually kind of cyclical. We see increases in the spring after we've terminated our cover crops, and it goes lower in the fall after the corn harvest. This is just to show it a little closer, and you can see how it's happening in our legumes versus our non-legumes or grass and brassica treatments. We are seeing that increase And because it's happening in the fallow, a lot of this is likely due to conversion to no-till production and just having that biomass accumulating on the soil surface each year. But we are seeing some different responses between legumes and grasses and brassicas. Sulfur was actually really interesting. You can see in our fallow, so we're not really adding anything to this. We had a spike the 1 year, but for the most part it's pretty consistent. But when we look at legumes and brassicas where we're having that influx of biomass in the spring when the cover crops are terminated, we're actually seeing increased production of this enzyme, which is assisting with release of organic sulfur into inorganic plant available forms. This is another one that was built off of that project because we kept seeing this and we were trying to determine what's happening to these nutrients when we do it. So we switched it a little bit. We planted some mixes, anything from plain just straight wheat up to we had a treatment of crimson clover, hairy vetch, and radish, to black oats, wheat, hairy vetch, and radish. This top line here, those are our weeds that were there. Now in general, all of our cover crops did a really good job of weed suppression, but when it came time for the end of the season, or sorry, for termination going into planting, those weeds are not breaking down. Most of these are forb type weeds at this location. This was all annual ryegrass. In fact, I had amazing stands of annual ryegrass at this second location. Where I had wheat, where I had cereal rye and black oats, I had almost no Italian ryegrass in my field. Where I had nothing planted, we had a lot of biomass left over, and it's just not breaking down and releasing those nutrients. And this is what it's doing for nitrogen. I know there's a lot on this slide. Essentially what we're looking at, this is ammonia nitrogen, and at 2 different locations, 2 different years, and you see ammonia is kind of all over the place. We're not seeing any discernible patterns in ammonia turnover in the soil. But this bottom one is nitrate. Again, 2 different years, 2 different locations, and what we find is we start to see this spike of nitrate availability about 6 weeks after we terminate our cover crops. So what we're looking at is how do we recommend the best time to do termination relative to nutrient availability if that's one of your goals from the cover crop. If you're wanting to use that cover crop as a nitrogen source, when do you need it for the crop, and count back from there versus when you would want to terminate. Now that doesn't take into account things like potential carryover and surface cover and all of that, but from a nutrient perspective, that's what we're looking at here. And that's really all I wanted to talk to you guys today about, although selfishly I'm going to plug, we are hosting a Southern Cover Crop Conference in February in Baton Rouge. You're all invited. Love to see you all there if you're interested in cover crops or if you just want to come visit Baton Rouge for a couple of days in February. It's much nicer in February than it is in the summer. But with that, if anyone has any questions, I'd be happy to answer them.
Any questions? I don't think Baton Rouge in February is probably going to be better than Stillwater in February for that. Just a guess.
Maybe. We do occasionally get ice, very rarely. But they shut everything down when that happens, so yeah. Nice little vacation. It's just a couple of days off. Oh,
no questions?
Cool. Thank you all.
Thank you, Dr. Lee. Appreciate it. Oh, you just take it to Tom that way. Okay, so next speaker set up here. Tom comes from the frozen purple north.
Damn Yankee.
Yankee. Came across Tom when I started doing talks for the Kansas NRCS and such. So Tom is retired Kansas state agronomist, and the NRCS recently retired, and now he's some kind of consultant that does stuff. But I want to bring Tom in because he's starting to do a large-scale project, been involved in several large-scale watershed projects, and wanted him to kind of share what he's been doing up there, looking at water quality, soil quality, and in those projects. And so with that, I'll let you do a full introduction to yourself, okay. Point at that, and we should have your slide deck. Your slide deck's not up yet.
Story of my life.
Yeah. And I'll stand up. over here when you're getting close to your time.
Okay.
You can introduce yourself a little further.
All right. Well, as like Brian said, my name's Tom Roth, and I am a retired NRCS employee. 30 years of field experience, and then the rest of that was at the state office. Got to spend some time as the CSP program manager. Sanity finally broke in, and I got away from that. Moved over to the Ecological Sciences Department and took that position as a state agronomist. Yeah, I gave them to him. Should be 2022, OSU Winter Crop School.
Don't have to wing it.
I'm good at that. That's one thing I've learned from this project. You know, one of the mottos of the Marine Corps, Force Recon, adapt, improvise, and overcome. We're going to talk about some of the really fun stuff that nobody ever tells you when you start something of this scope. I guess there's a backpack and it's in there. I think it's in the second compartment. Should be the second one. It'll be on a little. So I guess we go through some of the background. About 4 years, no, it's on a thumb drive. Hey, hallelujah. Okay. Hey, it even works. All right. So, like I said, my name's Tom Roth. I had to figure out something to do in retirement. My wife said I couldn't play pinochle, pitch, cribbage, or play dominoes for 4 hours in the afternoon. So I started I call it an agricultural and environmental consulting company. And this is a paired watershed study. It's up in by Clay Center, Kansas. That's about 30 miles from the Nebraska border. So the big question is, why are we here and what are we going to talk about? Well, one thing we want to talk about is to educate the audience, and that's mainly, certified crop advisors. I think there's some county extension people that are probably here. So we want to educate you about what NRCS is doing on-farm studies to evaluate water quality. And for some of you, you may be sitting there going, what in the world is NRCS doing research? Because you'll hear it more than once from some employees is we don't do research. Well, the Environmental Quality Incentives Program has what are called CEMAs. They're Conservation Evaluation Management Activities. So this particular, of the water quality portion of this project is funded through EQIP and using those CEMAs to install the equipment and then to pay for my time and all of the analysis that goes on of the water. We do some grain analysis, but that's not really that big a deal at this point. We're just mainly interested in what's in the water. And then the other thing that NRCS has decided to do is they're doing a very robust review of the Crete soil type. There's about 400,000 acres of Crete in Kansas and mainly Nebraska. And so what they're doing is looking at what they can do to update their physical properties. All right, so the big question is, why do we care? Well, some of you might remember in 2014 they had a rather significant algal bloom on Lake Erie. Basically shut down the water supplies from the intakes coming in. So you had to, basically they were living on bottled water at that time. And so Lake Afton, that's a public recreation lake up by the city of Wichita. Wichita is the largest city in Kansas, so that has a significant effect. And, you know, it has an effect because it can limit contact recreation. And once they get to a certain level, they'll say, we're going to close this portion of the lake. And this is in Kansas. And so in 2013 or 2020, we had 30 lakes that had confirmed HABs. And when you think about it, this is an 18-inch rainfall area. So it's pretty dry. And then this is what the surface looks like. Some of you may have been out on a lake and driven through one of those green slime pads, and you get to your boat ramp and you pull it out and it's just covered in a very nice green pea soup. So we'll call it that. So let's talk a little bit about what's the process that drives those harmful algal blooms. So we have nitrogen and phosphorus, and basically that increases our phytoplankton population. And that leads to an increase in the amount of those particular little critters. And we see those blooms, those large green pods. Sometimes you'll see them out just in like a farm pond, so like a stockwater pond. Sometimes you'll see that stockwater pond's got kind of a green sheen to it. So that's a small bloom even out in like a stockwater pond. And so those phytoplankton, they block the sunlight. And because the sunlight can't penetrate down, you know, a lot of our aquatic vegetation that grows below the water surface doesn't get that sunlight, and so they die. And when they die, they go down to the bottom of the lake or whatever water body we're dealing with, and they release more nutrients as they decompose. And so when we kind of look at what can be the potential sources of phosphorus and nitrogen, you know, one of them is land application of nutrients, fertilizer. Another source is your basic self-cleaning feedlot. So whenever you have a nice rainfall event, cleans that feedlot right out. Here, this is a wastewater discharge. Anybody take a real nice educated guess where the discharge point is? It's pretty nice and clean up here, and there's a lot of your filamentation algae and down here. Another one that I don't think a lot of people think about are failing septic systems. So sometimes we'll see these systems that are failing, and so they're bubbling raw sewage clean up to the soil surface, right on top of the ground. So our research objectives, and again, I realize some of you may be stunned that NRCS is doing research. That's okay. We want to look at water quality, and specifically what we're looking at is phosphorus impairment. And then the other thing that we're looking at is helping the soil survey crew measure soil water, and also they're going to be measuring dynamic soil properties. And we'll get to those dynamic soil properties a little later in the presentation. So here's our watershed. This is out in a cropland field. We have 4 sites out there. And you can see they aren't terribly big. I mean, we go from 2.4 to 3.2. acres in size. And where the little red dots are, that's where our samplers are located. And so our crop rotation, and I probably should have changed this a little bit because it's grain sorghum / corn, because phosphorus application is very similar on a per bushel basis for those two crops. And then last year was soybeans, and it's one of the first soybean harvests I've seen in 27 years I've been up there. We averaged probably 30 for this field, and last year it was probably closer to 55 or 60. So it was, and we'll show the rainfall pattern and temperature pattern a little bit later in the presentation. And then this year we're doing wheat, double crop beans, and hope that it rains after the beans go underground. And so we have the four watersheds. The top watershed, we're doing build and maintain phosphorus application. We're going to be using a cover crop, and we're going to go cheap. We're just going to plant wheat, just so we don't have to argue with him about what he wants to do. Because he said he's always got wheat around, so it's just really easy. As soon as crop comes out, we'll drill it to wheat, and then terminate that in the spring. And we're also going to be doing, on this one, we're going to be doing 2x2 row placement to see what effect that has on our runoff, how much phosphorus we may expect to see in the runoff, how much sediment we might expect to see. The next one is build and maintain with no cover crop, and that's going to be a surface broadcast. And then we're going to do a sufficiency application, surface broadcast, and then the last watershed will be how he's doing everything at the current time. So this is Benson Farms. Ron Benson's the owner and operator. His brother-in-law is his consultant, and they do they're really into technology. So they grid sample every 3 years, and they have harvest yield monitor data that they use to develop their prescriptions. They also use, I mean, Ron's pretty, he's tricked out. I mean, he's got down pressure sensors on the planter, and it's fun to just ride in the cab with him. So it's really enjoyable. He usually surface broadcasts late in the winter, early in the spring, and then he also does uses the Encirca service to determine what his nitrogen side dress would be for corn. I wouldn't say it's contoured, it's more cross slope. He goes across the slope on the field, and he currently doesn't plant any cover crops. So the two, excuse me, the two fertilizer application strategies are ones that probably maybe get used down in Oklahoma. I'm pretty sure Build and Maintain does. I know Brian's talked about sufficiency before when we've had him up to Manhattan to talk about fertilizer application strategies. And so sufficiency, basically we're going to maximize profit in that given year. And then we want to minimize the nutrient application and fertilizer costs at the same time. And of the two management strategies that we're going to employ, this will probably lead to the lowest application rate. Now, having said that, I'm not sure we're ever going to apply fertilizer to that particular area. Because right now it's sitting at 43 parts per million P. And so I don't know if we can ever draw it down to where we're going to hit that threshold for a sufficiency application. But that's the, when we submitted it to NRCS, we said, here's what we're going to do. And they said, okay. So we'll see. A couple of years of 200 bushel corn might help us, but I don't know if it'll actually draw that much down. Build and maintain, that's one that's been around for quite a while too. You know, we have a threshold value that we want to get to, and we have a time frame that we're going to use to build that up to. And so we look at that, what that threshold is, and the number of years that we're going to be employing that build strategy, and usually leads to a little bit lower rate spaced out over time. Once we get to that soil test level that we're shooting for, that threshold value, then we're doing what Kansas calls a maintenance application. So what we're doing is looking at what crop removal is and then putting that in back as a nutrient application. And that's where we hope our stuff that we're collecting on the grain, phosphorus content, can compare that to what K-State's using now. Some of the stuff we're trying to do, we're going to turn and let K-State have, and hopefully maybe they can use it in some of their work. And if you have a question during it, if you got an empty water bottle, throw it. I'm not as spry as I once was, so dodging moving objects can be somewhat of a challenge for me. All right, so this is the one that I think has some things going on that are going to make it pretty interesting. So the first thing we're doing is we're going to stay with that build and maintain strategy. So again, we have a target, we have a threshold value we want to hit. And we have a specific time frame, number of years that we're going to use to get there. Next, we're going to be planting a cover crop. And that cover crop gets planted after corn, sorghum, and after double crop beans. Because as soon as the beans are the full season beans get harvested, we're planting wheat right after that. So we're going to do fertilizer placement. We're going to use a 2x2 row scheme on that. And what we think we will see, we don't know for sure because we're not far enough into it, is we'll see lower total suspended solids and dissolved reactive phosphorus. And we base that thought on some work that they've been doing at Kansas State University. Kansas State has they call it the Kansas Agricultural Watershed classroom. And they've been doing some of this since 2015, I think. So they've gone through a 5-year cycle, ran out of money, got some more grant money, off and ran it again. And we also expect to see less dissolved reactive phosphorus and total phosphorus using our placement system. So this is the study I was referring to, and the long title is Decreasing Dissolved Phosphorus Loss, and we're building on soil health and multi-purpose conservation systems. Nathan Nelson is the professor, and he teaches the basic soil fertility course at Kansas State University. Peter Tomlinson is on the extension staff, and he's their environmental specialist. Craig Rosenboom is a cropping systems specialist. Elizabeth Yeager was a PhD student. Deanne Presley is the soil specialist. And Ganja Hederachi is a professor there, and Gerard Klutenberg, we call him the notorious Dr. K. He has soil physics is his main area, so soil water. And he's going to be doing some calibration work for us. We've got some sensors, and I'll try not to get too far into your soil sensor presentation. I'll try and stay away from that. It's the part of it that I was most nervous because I knew he was going to be here. And so they've had this thing for 5 years. They got funding for another 5. So they're moving around and doing some pretty neat stuff. So based on that K-State study, we're going to expect to see somewhere between a 5 and a 50% decrease in dissolved reactive phosphorus by using that spring placement method. That's what they head for a reduction, so we're hopeful that we'll see that same number. And then this is the total phosphorus. So the total phosphorus loss here, and 19 was a pretty wet year for us, but we can see that now the control, that's a no phosphorus application. And then fall broadcast is the red bar, and spring injected is the blue bar. The phosphorus source on that, I think it's 1846. Probably 1034 injected, but their surface broadcast is 1846. This one's not my study. I'm not a university professor, I'm not a university researcher. I work for Benson Farms. That's who I work for. I'll disclaim it a little bit. I did ask him what I said, so what's the control? And he goes, 0. And I go, okay, good. That's what I thought it was, but, and I can sure find out for you if you really want to know. All right, so data collection. Water quality, we're measuring total nitrate, total nitrogen, nitrate nitrogen, and nitrite nitrogen. Phosphorus is total phosphorus and dissolved reactive phosphorus. And then we're also measuring total suspended solids. On the soil side, they are measuring infiltration, water content, but I think I'm going to steal somebody's, I'm going to start calling it water climate or soil climate. I like that. I like it a lot. And dynamic soil properties. So this is a setup. This is what we use to collect our samples. So the terraces, and each one of those watersheds is basically a terrace interval. And so the terraces drain into these flumes here. Now, the flume is actually just this little portion of it. The rest of this is called the approach. So there's about a 4-inch overfall right here at the end. And so to settle that water so you don't have a lot of turbulence and get a fairly uniform flow across the sampler, or across the flume, it runs across that approach. So in order for us to get samples, 2 things have to happen. It has to be 0.015 meters deep, so 1.5 centimeters, and we have to have a specific flow volume go through that flume. Now, once that flow volume goes through and the depth's maintained, then we start drawing samples. So this is the sampler. It's refrigerated. It's nice in the summertime. Even if it doesn't rain, I have to go out there once a week, stick something in there the week before you, and then next week you go out, you got a cold beverage. I'll get to this a little bit later, but this is actually what makes that whole system work. This is a bubbler flow module. We use deep cycle marine batteries for our power source. And then, believe it or not, that's a heater. So we have to heat those huts because we have to be able to collect samples, runoff samples, year-round. And in the wintertime, we're kind of sitting there going, we don't think it's ever going to happen, but they said, you're going to install heaters, and we said, you bet. Originally, they wanted this whole thing covered and a heater underneath it. And we were sitting there trying to figure out, why would you make us do that? And these are flow-weighted composites. So there's a 2.5 liter jug inside this sampler. You can see the spare one right there. And what it does is when it meets the flow parameter and the depth parameter, then it draws a 200 milliliter sample. And it'll just keep drawing them as long as you have the amount of flow and the depths maintained. It'll just keep sampling until it fills the sample, fills that jug up, if you get that big enough of rainfall event. And then when you go out, you see this big error. You have your program defaulted while you were gone, and you're sitting there hoping that it's still working right. And so this is this is a little stainless steel tube. And here in this bubbler flow module, there's a little compressor and there's a pressure transducer. And so it's always sending out little impulses of air that come out here, travel across, and then come down here, and there's a little stilling well right there. And so if there's more water in there, then it tells the computer up here on the sampler, I've got flow. And it converts that to, I've got depth, and then it converts, the computer program converts it to flow. Now, what they don't tell you when you buy one of these, that one's about $6,500. It's worthless without that. It won't do anything. And we missed a significant amount of rainfall events the first year because we had to wait for them to ship those to us. Those are only $3,300. And we had 3 out of the 4 that I had to send back because they didn't work right. All right, so we have power. How do we get power to it? Well, I was sitting out at Ron's shop Monday afternoon, and we were sitting there going, you know what we should have done? We should have taken a bunch of this money and just trenched electricity up there and been done with it. Because it's the waterway outlets at his bins. So we could have tied right into his power supply there at the bins, ran buried conduit, done the whole 9 yards. But we get our power supply, we have 2 90-volt solar panels. And so this is what powers the sampler. This is our climate station. We have 2 of those climate stations. And it also has 2 13-volt deep cycle marine batteries that the solar panel charges. Rain gauge, anemometer, I think this is our pyrometer, relative humidity, and barometric pressure. And then there's 2 moisture sensors buried in that waterway at 20 and 50 centimeters. So a little bit about the climate. Last year we had 60 rainfall events from October 16th, and we harvested grain sorghum on the 15th. And up to October 15th of 2022, and I was just late pulling our climate data off the stations. I think Ron harvested on the 9th, but we didn't have any rainfall events. So of those 60, 25 of them were less than 1/10 of an inch. The biggest one we had, which was interesting, because it was the one time I decided I would leave and go do something. And the 2.8, that was on May 26th. And Ron was actually out there on some of the stations with jugs holding them underneath the outlet of the flume to catch samples. Actually, I think it was his son's, but he said it was him. So March, May, and July, we exceeded our long-term average. And all the other months were below average. Our 30-year average up here at Clay Center is 29.9 inches. As of October 15th, we had 2365, so that gave us a 6 1/4 inch deficit for the growing season. So this is just a graph of our precip. So you can see here, we barely eked it out in March, just barely got above the 30-year average. May, it was a lot higher. Most of that came over that May 24th through the 26th. We had some fairly sustained rainfall. And then again in July, we had 3 days where we had pretty much continuous rain for those 3 days. And then we're just getting nothing right now. So this is a hydrograph. We've got depth over here, time across the bottom. So we've got 1, 2, 3, and 4. And it corresponds, you can see, so this was the 24th through the 25th. Look at this. Whoa, I got to roll. 24th through the 25th, and the dashed line is that depth, that 0.015 meters. Similar, I think the interesting thing is, notice how this long discharge curve. You know, we get pretty rapid to here, and then we've got kind of a really long gradual discharge curve. So this is part of that KSU study, and these are their cover crop effects. Here's your runoff numbers, and cover and no cover. And again, so this was that previous slide. This is their discharge curve. So you can see where they get a peak discharge of about 1.4, and that's no cover. With the cover, right about 1. So you have a lower peak discharge, but you've got a longer discharge time. So it takes longer for it to get out of that field. So here's our runoff numbers. 3, which is our smallest one, had the largest runoff. And this is in inches, so 2 3/4 inches. 4 was about 2. Just, 2, about 2.75, and then just right at about 2.5. Now, we haven't had any cover crops planted out there, so it'll be interesting to compare that base. Here's our temperatures. So we're above average November, December, January, February. We run just through, about April, a little bit below. This one's interesting. That was our Colorado and Kansas for about a week. It's about 75 degrees during the day and just about low 50s at night. All right, total suspended solids. Why do we care? Well, what's bound to that soil particle when it erodes out of the field? We've got phosphorus bound to it. And so, and this isn't, when I finally got to looking at it, it's not really surprising why this one is the highest, because we had to do quite a bit of work to get the terraces so that we had enough storage in the ends of the terrace. Basically, the terraces then become a tile outlet terrace because you've got that discharge area at the end. So we did quite a bit of work getting those terraces built back up, and I think we just caught a lot of sediment from that. And so based on the KSU study, they did see quite a pretty pronounced effect in reduction. Here's your cover crop discharge, and there's the no cover discharge. So you've got less sediment coming out here. So we have one of the parameters that we're looking at was nitrogen. Nitrogen is a component of harmful algal blooms. So this is total nitrogen. And, 3 and 4, really all these are roughly the same. We're losing, we had about 2 pounds per acre, between 1 3/4 and 2.5 pounds per acre. So here's our nitrate load. Pretty small numbers. Nitrate, it gets even smaller. And again, real 2, 3, and 4 were pretty similar. Really don't know why at this point. We'll just have to see as the study progresses what happens. So here's nitrite. Interestingly, 3 was the bottom here, but 4 and 1 were roughly the same. So this is some sampling work that Dr. Nelson did, and he got basically they were looking at different sampling strategies, and this is a group that he's with across the United States. So we had 83 parts per million here. Phosphorus loading, pretty similar, dissolved reactive. 2 had the most. Here's the cover crop effects from K-State. 40% reduction in dissolved reactive with cover crops, and total phosphorus on that side. So why are we looking at soils? Well, the Northern Great Plains Soil Survey Office is updating their effects on the soils. So one of the ways we measure infiltration is we're using what's called a Saturo dual head infiltrometer. Soil scientists get to go out and spend a lot of time sitting on the back of a pickup waiting for water to go in. Basically, this runs at 2 different pressure settings and maintains the water level at a specific amount. And this is pretty much what you would expect a classical infiltration curve to look like a little bit. This is the flux, and then this is a weighted average of that. We use a Campbell Scientific 655, meter Teros 12, a Clima TDR 315, Campbell Scientific Soil View 10, and the meter group. And I'll kind of skip through the equipment part because maybe Tyler's going to cover some of that at this point. Okay. Well, I'm also running out of time, so I got to figure out what to give that. Basically, there's 2 different types of sensors we're using. One, which is the Campbell Scientific, and they're all measuring volumetric water content, bulk soil EC. They use dielectric permittivity and measuring permittivity and temperature. The Teros 12 works very similar. So most of these work using this system, dielectric permittivity. True TDR. So here's the one that's different. This is measuring water potential. Basically, it's a clay disc that you stick into the side of your area where you're installing them, and it measures the volumetric water content of the disc, and it uses a water retention curve equation to come up with volumetric water. Mice love these systems. Originally, this was just black plastic wrap going up. And we were out there while they were installing it, and we go, you want to put that in conduit or you want to do something with it? You're going to have more mouse problems. Basically, they lost the very top terrace because the mice got in there and kind of chewed it up. So this is what our graph would look like. Now, the other thing is, you're looking here and going, what's this mean? You're having more than basically that's saturated. Well, it wasn't saturated. So you need to do a calibration for each one of these, for some of these sensors. So we go out, take samples, put those into a tube, wet them to a known water content, and then you just start measuring. Dynamic soil properties. So dynamic soil properties are those that can change with either land use or management. And some of you may have heard of the Kellogg Soil Lab up at Lincoln, Nebraska. They're going to be doing all of these tests. So these are some that I just threw out. I've asked, emailed them several times, what are you going to do? What are you going to measure? Can we find out? And I know it's not part of our study, but it's nice to relay this information out to people. And so bulk density, well, that's just a mass per unit volume of soil. And so we just take the weight divided by the volume. We get 1.3 grams per cubic centimeter. And then they did a microsoil survey out there. So basically they soil surveyed 11 points and did a full soils description for each one of those cores. So what's next? Well, we're still going to be collecting soil water data. Hopefully Lincoln's going to give us their list of what they're going to do for dynamic soil properties. We have one more year of his nutrient management, and we'll shift to our design at the end of 2023. And so I'd like to thank the following people. Benson Farms LLC, and that's Ron, Catherine, Alex, and Jude. John Straub, I couldn't figure out a good title for him. He works for them. He's a mechanical guru. I mean, if I'd have something go wrong and I could go pick his brain about what do you think's going on here, and he would help me. Elliot Carver replaced me at the state office in Salina. Jay Roth, no relation, is a environmental engineer for NRCS at Portland. Something that we've had interest in from Kansas Department of Health and Environment and the Army Corps of Engineers is their Silver Jacket program. So they're trying to figure out if they can use soil health, cover crops, these kinds of things to minimize flood risk. And the people that I would be lost without over at Kansas State, Dr. Nathan Nelson, Sarah Rogowitz, who is his research assistant, Dr. Gerard Klutenberg, and the rock stars of the whole thing, which is the soil testing lab at Kansas State University. I will do my best to entertain questions at this time.
Don't have time for questions. So
see, that's the way you do it.
Tom. Thank you, sir.
Thank you.
Nice to be coming up. All right, appreciate it. So, see you around for the poster session. We'll have our next presenter coming up, Dr. Tyson Ochsner. Then remember after this,
Thanks, sir.
come back up here. I'll request judges for the poster session. I need at least 12 to come up before the meeting. If you get a Visa gift card if you help me out. But with that, our next speaker is Dr. Tyson Ochsner, the smartest man in our department. So I'm glad to have him out here and share some of his work. He does some crazy stuff. I just want to say that. It is crazy, but crazy awesome. Thank you, Tyson.
Thanks, Brian. Wow, that's quite an introduction right there. If I was the smartest guy in the department, I probably wouldn't have been out digging holes this morning. I would have been in here for the lunch or something. Great to see you all today. Good to see some old friends in the room. I'm Tyson Ochsner. I grew up down in Tillman County, Chattanooga. I went to school down there, and I grew up on a wheat and cotton farm down there. And went to school here at OSU. Was working out of the state for about 10 years, and then have been back here since 2008. and I get to do teaching and research related to soil and water, which I love doing. Love working with the students and working with our cooperators. And so I want to just tell you about one of our current projects right now. And I'll just say, feel free to interrupt, okay? So we don't need to wait till the end. I think there'll be plenty of time for discussion. I wasn't planning to talk the whole time, so I'm hoping you all are planning to talk some. This project is called Chronos, and I'll explain that abbreviation here in a minute. But what we want to try to design are systems that are optimized for monitoring the soil, the plants, and the atmosphere in a cropland environment, because those systems don't currently exist. And so that's what we're trying to develop. And so just a little context for that. So producers, consultants, researchers like myself, many of us could benefit if we had better sources of information about the current conditions of our soil. How much water is stored in that soil out in our cropland? How's my crop doing? What's its percent canopy cover at this point of the year versus last year or prior years? And then what's happening with the local atmospheric conditions, especially, of course, the local rainfall, which varies so much from place to place. If we had better information like that, I think we could probably make better management decisions. And I'm mostly focused here about like mid-season management decisions. For example, how much am I going to side dress? How much do I want to invest in this crop moving forward? Might help us to better predict our crop yields mid-season. For our researchers, it would help us improve and evaluate our growth models. And a lot of this work, we try to do it with remote sensing now, and remote sensing is awesome, except it's just all guesswork unless you've got ground truth data, right? You're just guessing. So we try to develop better remote sensing algorithms if we can first get better ground truth data. But here's where the rubber really meets the road, and Tom's talk was a perfect example for this. Most of our soil moisture monitoring networks, we don't put the station out in the middle of cropland, right? Because if you do, mice are going to be the least of your problems. Your biggest problem is going to have green paint on it, right? It's tillage, it's planting, it's spraying. All the agricultural operations are going to destroy the equipment. And trust me, I've tried it, okay? It's just real hard to coordinate that. The first point I want to get for you here is just so that you understand that it matters. Okay, if we have our monitoring station on the edge of your field, but not in the field, that's not helping us out very much. That's not a cropland station. It's not a cropland station unless it's in the crop field, okay? So here is, we have one of the best networks in the world in the Oklahoma Mesonet, thanks to many folks that have gone before us and done this work. And so the black triangles here show some of the locations of the Mesonet stations. And then the two colors on the map show our winter wheat cropland and then our grassland and pasture. I just pulled out those two land covers to show you. And you might look in first and say, well, okay, we've got some of our stations here in our cropland areas. But if you zoom in, not one of those stations is in a crop field, right? Anybody ever seen a Mesonet station in a crop field? If you have, tell me. It was an accident. It shouldn't have been there. Something happened. And so, this is why it makes such a big difference. These lines could not be any more opposite for each other. We measured this right here at Stillwater. So this is the ground cover. This is just the percent green canopy cover. So if you have full green canopy cover, that's 100%. You got dormant Bermuda grass, that's 0. Okay? And so this is comparing that Mesonet station, which is the black circles here, that's this line, its green canopy cover, versus a winter wheat field that's just 20 yards away. Just right beside it. Okay? They're directly opposite of each other, right? Here the grass is all dormant and dead. There's no canopy cover, and we've got 80 to 100% in the winter wheat during, February through April. And then come April, the grass starts to green up and get up to 100% canopy cover. But at that same time, starting about the 1st of May, the wheat starts to senesce, right? Well, that's going to make a big difference on how those plants are using water, isn't it? And that's going to make a big difference on the soil moisture conditions. It's going to make a huge difference. Okay, and we don't have any way of accounting for that. There is not any remote sensing product yet that can account for that. There's not any monitoring network yet that can account for that. So we've got to come up with something. Just to show you the impact, so we went out to a lot of farms, sampled soil moisture under wheat different times during the growing season, and then just across the state, and then we went and used that data to develop a model that could pretty effectively predict the soil water content under winter wheat if we knew some of the key parameters. Okay, and then we ran that model for a large number of locations across these different districts of Oklahoma. So South Central, North Central, Northeast Oklahoma, the Panhandle, and then we ran a lot of years, I don't remember how many years, 18 years of simulations of soil water content. Okay, and then we just plotted the median. That's the value above which or below which 50% of the values fall. It's about like the average. Okay, so you can think of it as the average. And that's this solid black line. So you can see it typically peaks early in the year. So day 60, that'd be about 1st of March. Okay, somewhere in there. Somewhere in there we have a peak in our soil moisture under wheat, and then it starts to decline, reaching minimum values. Typically, it varies from sometime in April, that's around day 100 here, out getting closer to day 150 in the Panhandle. That'd be about the end of May. All right, because of different climates across the state. That's how the winter wheat dries down the soil moisture conditions. And that's totally out of phase with our grass, right? So the grass, you can see it's peaking later. It's peaking in maybe April or May, maximum soil moisture conditions. And this minimum is coming out here in July or August or something like that, right? So these two systems are totally out of phase, and yet this dashed line is the one that we monitor, and the solid line is the one we want to know for our cropland decisions. So we've got a real problem. If you're not convinced yet, let me just show you this picture. This is one of my favorite graphs that we've ever produced out of my research program. And this was by Dr. Patrick Nani, who's now with Tom and others up there in Kansas. So this is Lahoma. How many of you folks have driven through Lahoma lately? There's got to be some. I got like 2 hands. That means the rest, I got 50 people in here asleep. So anyway, there's Lahoma. And there's the Mesonet station outside Lahoma right there, that little red arrow. And that's just an aerial photograph. And you can see that, the mile sections here in the fields. And this is just converting that land cover just to either grassland or cropland. And here we're assuming that, grassland or winter wheat cropland. Okay, so the brown is the winter wheat and the green is the grassland. And then we ran our model for every single point on that map to estimate the soil water that would be under grass and what would be under wheat. And then we intersected those two together. And this is our best estimate on this day. This was October 15th, 2011. We didn't know what we were in for at that time, but 2011, October 15th, 2011, look at the contrast in the soil moisture conditions. Which one of them is here at the bottom where the plant available water is near 0? Which land cover is that? It's the grass. There's almost no available water under the grassland on October 15th, 2011. But that wheat cropland right beside it is almost full of plant available water. It's almost full. You got to see how dramatic that difference can be. It can be really dramatic. And I've got a question here on the slide. Can anybody tell me what happened with the 2012 wheat crop in Oklahoma? Good year, bad year? Pretty good year. Right in the middle of that intense drought from fall 2010 up to May of 2015, we were in intense drought, but we pulled out a really nice wheat crop in 2012. Why? That's a big part of the reason right there. We started with a really nice profile of water in 2012. But nothing could measure that other than you walking out there with a shovel. We don't have any monitoring network. We don't have any remote sensing that can pick that up. Okay, so we're trying to work on that. So we're working on this project with the folks at K-State, cropland observatory nodes, and you take some creative liberties with your acronym and you come up with Chronos. And so that's what we're calling them. And this is Cole, and Cole is here today, and he's presenting a poster on this. And so anything that I'm unclear about, go check with Cole right after this talk. So we're developing these stations. They are portable, in quotation marks, stations to monitor soil moisture, green canopy cover, and atmospheric conditions. We're trying to get them portable, and we're not there yet, if I can just be honest with you. Because there's nothing in the ground, okay? We're measuring soil moisture without putting anything in the ground. I'll explain that in a second. But you got to be about coal size to move that yourself, okay? So I'll hurt my back if I'm trying to move that station by myself. It's not really as portable as we want it to be yet. We're going to keep working to get them smaller and more portable than what they are right now. So we're headed that direction. So they're not as portable yet as what we want it to be. And this, you all can maybe give me some feedback here, but our thought was we make them portable enough when you're doing your field operations, you get out of the tractor and just move that thing over 15 feet and then continue on your way. Well, what do you think? How do you think that worked for us? No. No, that didn't happen. Farmers, our cooperators were just going around them, whatever. Well, if you just go around it, then that area's not representative anymore. We can't have that. Okay, so we thought, I know what we'll do. We'll make them fold down. So we actually cut these things into, we put a hinge right there in the middle and designed it so you go out there and pull a pin and just lay it over on the ground. You can take your spray boom right across the top of it. You think that worked? No. No, because nobody's getting out of the cab. They're not going to get out of the cab. So that station is going to have to get out of its way by itself. I think that's the direction that we're going to have to head. It's going to have to be something that will move itself out of your way.
So how come you're not putting a tensiometer in the ground?
Great question. Now, how come we're not putting a tensiometer in the ground? Because once I do that, I can't make it portable.
Sure you can. Hydraulic cylinder.
Yeah, okay, all right, maybe so. Maybe I need to make it bigger and beefier with machinery to insert it and pull it out. I like the idea. Let's talk some more about it. I'll show you why we don't need to here in just a second. Okay, so our objective is to evaluate the performance of these things on 3 working farms during the winter wheat growing season. And so we got cooperators down there at Chattanooga. Aaron Henson, I think, is here and helped us get a cooperator there in Tillman County. And then had one on the station, on Dr. Arnell's station there at Chickasha. And worked with Patrick Curl up there by Bremen and put out these stations. And so here's what they look like. So on top we have an all-in-one weather station. This thing is something it's available commercially, and so we took that sort of off the shelf and started to use it. It's a really nice product. Out here on the arm we have our camera. So we're using a digital camera to take photos of the crop multiple times a day. So it's facing down at the canopy. It's a trail cam with a cellular modem built into it. And then this is our soil moisture sensor down here. It's a neutron detector. And I'll explain how that works. And so we integrated these into a seamless package. They collect the data. They send the data out by the cellular network and they send it to our servers here where we can process it. Okay, this is the wild wacky stuff, or some of it, that Dr. Arnell was mentioning. So we're actually using, instead of a tensiometer, we're using cosmic ray neutrons to measure the soil moisture without touching the soil. Okay, so here's what's happening, and I didn't know about this until about 10 years ago. I started learning about this. But so there are always cosmic rays coming into Earth from outer space all the time. Okay, and they're mostly high energy protons. I'm not going to get too much into chemistry here, but you know, a proton is the little positively charged part of an atom. Okay, so they're mostly high energy protons. If they come into the atmosphere and they smack into something, nitrogen or some other molecule in the atmosphere, and if they hit the nucleus of that atom, they'll cause it to it'll knock a neutron out, right? I was going to tell a neutron joke, but I'll save that ask me later if you want to hear my neutron joke, okay? But if they knock out a neutron, it doesn't have any charge on it, right? It's an uncharged particle, and it starts flying out at a high rate of speed, and we're talking millions of miles per hour. Okay, so this thing's flying, okay? And so it's flying down towards the Earth, and that neutron may hit another molecule and knock out something else, okay? And so from there, we get this cascade of these neutrons coming down. The only thing that's going to really slow these neutrons down is if they hit something that weighs the same mass that they do. And so the thing that weighs the same mass, a neutron has a mass of 1, atomic mass of 1, so does hydrogen. So hydrogen has a mass of 1. So if this thing hits hydrogen, it'll get slowed down. It'll get its energy split in half, basically, and slow it down. So if we go out into your cropland, where's the hydrogen? Well, it's mostly in the soil water. That's where it is. And so the more soil water we have, the fewer of these fast neutrons are going to be moving around above the land surface. And so we put this device out there and we just detect the neutrons. And they are representing the soil moisture for about the top 1 foot depth over an area that covers about 30 acres. Okay, with one sensor. So that's what we like about this technology. It's non-contact and it can represent a field size area. All right, trail cam, I think you understand that. We're taking pictures multiple times a day. Those are customizable. And then we analyze that picture to determine what's green canopy cover. Okay, so we have a algorithm called Canopeo. Just out of curiosity, have you used the Canopeo app at all? I've got a few users, okay, so use it if you're not, it can be helpful. But we're using the Canopeo algorithm to figure out how much green canopy cover we have, and then we use the all-in-one weather station, which I'm going to skip over. So we validated it. We went out and took soil and vegetation samples to validate our data. We also compared with the mesonet stations at Chickasha, Granfield, and Blackwell. So at Chickasha we're about 100 yards away. These other two are about 10 miles away from where we were working. And we looked at the reliability as well. Okay, here are the data. So soil water content on the y-axis here, and then the whole winter wheat season on the x-axis. And the blue dots are what we measured with these chronostations. And the red symbols are what we measured by going out to those wheat fields and soil sampling. And it did really well for monitoring the soil water content. We thought, this would be interesting. Let's see what the mesonet would have told us. So at Chickasha, that mesonet station about 100 yards away, that pink line is what it would have told us for soil moisture. Not even close. Chattanooga, same story. We were really far off if we would have used the mesonet. But that station is 10 miles away, to give it some credit. And this one really surprised me. This is one of the most surprising things I've seen in this project. This mesonet station at Blackwell tracked almost exactly with that wheat cropland over at Bremen. I can't tell you why. That was a real surprise to me. Anyway, that's what happened. Truth is stranger than fiction sometimes. That's just comparing what we measure with the sensor versus our sampling. Looks really good. We're within about 2% by volume. This is the green canopy cover. And so you can see here that canopy cover is pretty stagnant during December, January, February kind of time frame. Not much happening. Bremen, we had very little plant cover. We had more at Chickasha and at Chattanooga. This one at Chattanooga was actually being grazed. And then you come into March, and that's when you start to see the canopy really develop. Again, the blue dots are what we measured with our cameras, and the red dots are what we measured in the surrounding field. And we did okay at Chickasha and Chattanooga, but we were pretty far off at Bremen. The site where we put the station just ended up not being very representative of the whole field. And so we had a mismatch there. And so that's, again, the 1-to-1 plot. It's not very good. We were off by 10% on average. So that's I think, unsatisfactory. And I'll tell you what we're going to try to do about that. Last one, though, is just comparing the mesonet. So this one's confusing. The agreement is so good here you almost don't believe it. But the mesonet here is the red or blue line, and what we measure with the Chronos is the red or blue dot. They're indistinguishable. They're just right on top of each other for the most part. So that's working really well. The weather data is good. Okay, I hate to show tables, but I'm going to show you this one real quickly. I'll just show you what to look at. We found that with rainfall, precipitation, P, this statistic is a Nash-Sutcliffe efficiency. We want this to equal 1 for perfect agreement. And we were pretty good at Chickasha. We were about 0.87. You can pass my class with a grade like that. It's not bad. But you can actually pass with other grades too. But at Chattanooga and Bremen, we were a little bit off. That could just be by distance, but we were off in one direction. This station was actually shooting a little bit low. It seems to maybe underestimate precip a little bit. So we need to look into that further. Another thing, the temperatures, of course, that's just excellent. So no problems there. Solar radiation was pretty good, but again, we're shooting low, 10 or 12% on the radiation. And so Cole's been corresponding with the manufacturers about that. I think there may be some corrections coming there. Pressure was great. That's easy to measure. Wind speed also, we were a little bit high on wind speed. And we're still trying to figure out exactly why our wind speed was a little bit high. But here's the biggest problem we had was with these cameras. And somebody come and tell me afterwards a better camera. We're using a camera called a barn owl camera. I don't know if anybody ever used those. We just had they missed a lot of pictures. We had about 20% of our pictures that were just totally, they didn't happen. The camera never took the picture or never sent it. And then we had about another 20% that it took a picture, but we couldn't use it for green canopy cover analysis. Maybe it was blurry or something. There was something wrong with the picture. So our error rate for the cameras was really high. Everything else, our error rates were really low. So we got to figure out a better way to monitor that vegetation. And so that's where we come to our next steps. And so, and there's Cole working away, happy as a clam there with soil sampling. So Cole and our group is working to see if we can use, remember I talked about those fast neutrons? Okay, well guess what? There's also slow neutrons. Okay, just like there's slow people like me. So fast and slow, and the slow neutrons appear to be correlated with the biomass. So Cole's built a new version of the detector that can measure both the fast and the slow neutrons at the same time. So we're going to see if we can use those neutrons to measure how much biomass is in the field. Okay, so we'll get back to you next year maybe. So anyway, that's what we're up to with this project, and I'd love any suggestions like the one earlier about how we could make this better, because it's got to work on working farms or the whole thing's a bust. So we got to make it practical. So anyway, thanks to all the folks, our Rain-Fed Agriculture Project that funded it, our cooperators, thanks to Dr. Arnell, Michael Pettyjohn, and all the students and folks that made it happen. Appreciate it.
One of the problems with your camera was it fogs on the down angle, right? So think about that. So that camera facing down fogs up because of the way, even up facing camera will fog on game cameras, right guys? So down facing made it that much worse.
Yeah.
So that's an issue with that camera.
Yep.
Any questions for Dr. Ochsner?
Why don't
We got lots of them, so why not?
we, why don't you use a green seeker?
Yeah, the question is why not use a, yeah, why not use a green seeker instead of the camera? So that's a possibility to use a green seeker or another NDVI sensor. The ones that we've looked at thus far, their footprint is even smaller than the camera. So the camera is getting about 6 square meters with its photo, and those sensors typically are quite a bit smaller than that. So I think it'd be a scaling problem.
Still on campus would be better.
Could, yeah, maybe so. Yeah, okay, yep.
Got next to Brian?
You're saying that neutrons fly to the hydrogen particle. Is that drive atmosphere moisture into the ground of the plant? Does that happen?
It's a great question. He's asking, does those collisions of those neutrons actually cause the moisture to move in or out of the ground? Not to my knowledge. I'm not aware that it actually is enough to move the molecules. But for the water vapor molecules, when they're impacted, it would at least affect their rotation. Yeah, so it would have some effect, but we've never been able to measure how it transfers it in or out of the ground. The neutrons themselves will go in and out of the ground. Yeah. The neutron is a tiny little, it's a piece, so the hydrogen atom has one proton, right, and one electron. Okay, and if we go to helium, I'm gonna get myself in trouble here. Then it's got a proton and a neutron. Okay, so it's a subatomic particle that has no charge, so it can get knocked out from the molecule because there's not an electrical charge holding it in there. So it's just a little teeny tiny piece of the universe. Great. You're thinking along the right lines. The lightning that we see, that is due to the transfer of electrons, which is another subatomic particle that's got that negative charge to it. So you're thinking on the right lines there. Yeah.
I was just curious about the cost of the sensor.
Yeah, okay, great question. Cost of the sensors, that's another thing we're trying to drive down. Our first generation sensors, which I showed you here, that entire package altogether is around the $20,000 mark, which is too high. The second generation, which we are now testing, is about half that. It's about $10,000. And our goal, hopefully by the end of the project, we've got a system that's about $5,000. That's what we're after.
Yeah, 4,500.
Any more questions? We have a few moments.
What about ammonia volatility versus soil and water?
Yeah.
You know, say like you just applied some nitrogen, is it going to be able to distinguish between the soil moisture and soil and the volatility of the fertilizer?
Yeah, that's a great question.
Or is it going to measure it the same?
That is a great question. If a person had a sensitive enough detector, maybe you could use the neutron detection to see some kind of signal from the ammonia.
Measure that instead of soil moisture at the same time.
If you think about the mass, so in the soil we might have, when that soil is full, it might have 100 millimeters. I'm sorry for the metric units, but that's about 100 millimeters of water in the top, in the root zone. Okay, that'd be about 100 kilograms per square meter of water. So if we take that up to a hectare, that's about 1,000,000 kilograms of water in a hectare. That's about 1,000,000 pounds per acre. And we're putting on about 100 pounds of ammonium, right? So you see why you'd need a really sensitive detector to be able to see that. But I love the idea. We should look into that. How did we estimate what?
I estimated the water content, the soil.
Yeah, we did that with modeling. So the model I didn't show the model, I'm sorry. I put the link to the paper there that describes the model and how we used it to make that map. We've only ever made that map for that small area because it takes a lot of computation to do that. Honestly, here's the thing about science for me. we're all living with these soil water patterns around us that are affecting our lives, they're affecting our pocketbooks, and nobody's ever seen them. We don't have a way to visualize those patterns, how that soil water varies across the landscape. We're not there yet. We're surrounded by these dynamic patterns. They're ebbing and flowing all the time. You know about them intuitively because you work on the land. But we don't have ways to map those out and show what's actually happening. We're trying to get there.
That's good, Tyson. Thank you very much. Give Tyson a hand again.